We are Harmony Science Academy students.
Of Harmony Public Schools.
Yes. And today we want to explain our PBL
project.
Yes.
So, essentially, what we've been working on for the past month is a fungus named the Pilobus. And basically what it does is it shoots its spores directly at light sources, such as this one up here. And what we basically did was we got this kit. We have Petri dishes. It came with Petri dishes with rabbit dung, came with cups, aluminum, waste bag, and of course the fungus itself. And we did our research and we figured out how it works before we worked on it. Also came with a scalpel. What we did was we used the scalpel to extract the Pilobus fungus, and we put it into the Petri dishes with the rabbit dung, and we let it sit there for a day, and in the fridge. And after that, what we did was we put it directly under a light source, and we saw how it reacted in that environment.
Yes. And after that, we figured we might as well test our theory, which was if it needs light source to grow. So basically, we used the cups. We covered some, we left one open, and we left one visible with light coming through. Basically, for 3 days, we let it grow. And every day we would compare the amount of spores we saw in the cup. And basically by the end of it, we came to the conclusion that it can grow without some light source, because it starts shooting, hoping it will spread anything that's nearby.
Yeah. It doesn't shoot directly at any light source because there are none present, so it just goes all over the place. But whenever there's a direct light source, it will shoot. Some will shoot off into other places, but for the most part, it'll shoot directly into the source. Our group was researching projects on the PBL website when we decided to study and analyze the Pilobus fungus, and did further research into the procedure of the project so that we could have a better understanding of how the fungus works.
On the first day we received our materials. We went over the procedures to get a better understanding of how we should start our studies.
On day 2, we used a scalpel to extract the fungus from the rabbit dung. Then we transported the dung into 2 agar dishes, and then we incubated them overnight.
On day 3, we then labeled the agar plates and made sure to keep the fungi under light source, upside down, and let it incubate for 2 more days.
After the incubation process, we removed the lids from the agar plates and we wrapped them in aluminum foil and created a hole in one and left the other covered. Then we placed the plates under a light source for about a week due to the Thanksgiving break.
We removed the aluminum foil from both plates once we came back and analyzed to see if any of the spores were still there. We then compared both plates to see how each plate was doing due to putting them in different environments. We recorded our findings by taking pictures of both cups, as well as taking more photos using the microscope. We proceeded to take more pictures and then did further analysis until we were satisfied with our test results. The Pilobus starts off as a black sporang and has been discharged onto plant substrate, such as grass.
A herbivorous animal from horses, cows, etc. then eats the substrate, unknowingly eats the sporangnium as well.
The sporangnium then survives the passage through a gastrointestinal tract without germinating and emerges from the excrement.
Once outside, spores within the subsporangium germinate and glow as a mycelium within the excrement, where it is a primary colonizer. Later, the fungus fruits to produce more spores, and the cycle repeats itself.
Thank you. Signing off from Harmony Science Academy.
Thanks for watching.